My name is Mark Tessier-Levine, and I have the privilege of serving as the president of Rockefeller University. We're gathered here this evening to celebrate the 10th annual presentation of the Meister Greengard Prize, an international award recognizing outstanding women in biomedical science. This is a remarkable milestone, and we're glad that you're all here to share it with us. Your presence is a splendid show of support for the wonderful prize established by Paul Greengard and his wife, Ursula von Rydingsvard, and other generous friends of the university. If this is your first Greengard Prize ceremony, I'd like to provide you with some background on this important award. As many of you know, in 2000, Paul Greengard received the Nobel Prize in Medicine for his crucial discoveries about the brain's biochemistry. Paul and Ursula decided to donate the monetary share of his Nobel Prize to create the Meister Greengard Prize for outstanding women scientists. A number of the university's friends and benefactors were so moved by Paul and Ursula's gesture that they also contributed to the endowment for this prize. Now, Paul believed, and continues to believe, that women aren't being recognized at a level commensurate with their scientific contributions. He was determined to use his new prominence as a Nobel laureate to address this issue through the creation of an international award for women scientists. Paul named the prize in memory of his mother, Meister Greengard, who tragically died giving birth to him. Now, over the past decade, the Meister Greengard Prize has honored women who have made contributions to many different fields, including cancer research, immunology, genetics, biophysics, and the study of memory. The discoveries of these women scientists are among the most remarkable made in the last 60 years by researchers of either gender. In conjunction with the 10th anniversary, Rockefeller's produced a commemorative booklet celebrating the 15 recipients of the prize to date. Here's the booklet. I invite you to read more about each of these trailblazing women in this publication, and if you didn't receive one, you can get one on your way out at the door. Now tonight, it's a special pleasure to honor Dr. Huda Zoghbi, the recipient of the 2013 Meister Greengard Prize. Dr. Zoghbi was chosen by the members of the selection committee. I'm honored to serve on this committee with some very distinguished scientists whose names you can find in the program. In recent years, Rockefeller has made it an institutional priority to close the gender gap in the sciences. So on this occasion, I'd like to recognize the Female Association of Clinicians, Educators, and Scientists, known as FACES, and the Women in Science Initiative. Each year, these two initiatives co-host a discussion with the Greengard Prize recipient and women scientists from our tri-institutional community. A new campus organization, WISER, Women in Science at Rockefeller, is also participating this year. These discussions have become a cherished annual event. Now, before I introduce our prize presenter, I thought you might enjoy a short film created by Paul and Ursula's daughter, Ursula von Rydingsvard Grieve. The film allows us to listen in as past recipients, presenters, and members of the Rockefeller community reflect on the meaning of the Meister Greengard Prize.
I think the Meister Greengard Prize is amazing. I think the story behind it is truly remarkable.
Professor Paul Greengard. Paul Greengard, upon winning his Nobel Prize, donated all of the money to set up a prize in honor of his mother, who died in childbirth, and to celebrate women scientists.
The 10 years have gone by very quickly. I've been delighted at how effective and how dramatic the response has been. The Rockefeller University has been wonderful. They've administered the prize. They've set up a committee of 10 scientists, many of whom are Nobel laureates, and other people have contributed to the prize. And I'd like to think that the Meister Greengard Prize has contributed to the greater equality being accorded women now. There's still a lot of discrimination, but it's much, much less than it was.
I think Rockefeller is one of the few institutions that has these programs explicitly meant to benefit and honor women in science. This prize, I think, is giving the Nobel Prize Committee ideas about who should they be thinking about next year. That myopia about, oh, there's no good women, goes away when you look at the list of winners from the Meister Greengard Prize.
Many women who received it told me that for them this was the most special award they received because it was given to them as a woman in recognition of their work as a woman in science.
It's in recognition of women who really probably often struggled for years and years and years to establish their own identity and to establish a career in a field that until very recently has largely been dominated by men.
The contribution that women can give to science can be very specific but very needed. Questions and interests that attract women to investigate are different than the research that men do. So maybe we will have a more comprehensive research because it will provide different perspectives and different views to questions that need answers.
It is difficult to manage juggling a career and a family, but it's possible. Prizes like this that recognise women in science undoubtedly inspire and encourage women to not only enter science but to stay in science.
And to the extent that women opt out of the most demanding pathways in science, of the most demanding autonomous academic and research careers, because they feel they can't do both, we lose extraordinary people.
Women are at least half of the humanity. To lose that potential, I think, is a terrible loss in all dimensions, but of course in science too.
It depends on your personality. You're not going to be making a discovery every week or even every month. You know, you mustn't get bored easily. I don't get bored easily. You must have patience. You must be able to do repetitive work with very little exciting reward. And then if something happens, it's magnificent.
If you're truly passionate about whatever it is you're going to work on, whatever it is you're doing, then all those other things, obstacles that get in the way, are irrelevant.
Events like the Greengard Award are great because they get us all excited. And I think that's what's so amazing, is that coming to an event like this, you feel it in the air how excited everyone is.
This year's winner is Huda Zoghbi. She's a great scientist, and I'm personally delighted that she was chosen to receive the award this year.
I got goosebumps when I got the letter. It's a great honor to get a prize, but to me the impact of the prize is far beyond that. To me the impact is a young scientist in a lab today. See that the honorees started just like them in a small lab, knowing very little, having no idea what they're doing, but they had a passion and something they're going after, that they will be inspired to do the same. And that's for me the power of this prize.
And now, I'd like to introduce our prize presenter, Ursula von Rydingsvard. As I mentioned earlier, she is one of the co-founders of the Meister Greengard Prize. In her own career, Ursula has also achieved international recognition as one of the foremost sculptors working today. Ursula is known for creating large-scale sculptures from cedar beams, which she cuts, assembles, and laminates in her studio in Bushwick, Brooklyn. Born to Polish and Ukrainian parents in Germany in 1942, Ursula and her family were among the displaced who, following the war, were forced to move from one refugee camp to another. They eventually left Europe and settled in the United States in 1950. A graduate of the University of New Hampshire and Columbia's Graduate School of the Arts, Ursula has taught at Yale University, and she began working in Brooklyn in 1981. Today, her sculpture is included in numerous permanent collections, including those of the Metropolitan Museum of Art, the Museum of Modern Art, the Whitney Museum of American Art, and the National Museum of Women in the Arts in Washington, D.C. As many of you know, in September, Ursula's latest work, Ona, was unveiled at the entrance to Barclays Center in Brooklyn. Its central location permits viewers to interact with the pieces Ursula intended, communicating with the massive bronze sculpture by touch. And if you do decide to visit Ona there, you might have time to pop in for a Nets game or a rock concert at Barclays as well. Now, I'm honored to welcome Ursula von Rydingsvard to the stage. Please, Ursula, if you'd join me now.
Thank you so much. I'm so happy to be here. I'm going to show you my work that I've done for maybe the last 6 or 7 years, but I'll start with one that's a little bit earlier. And I'm going to emphasize a little bit the process by which some of these pieces were done. So I'm going to begin with a piece that I did in Sebastopol, which is about an hour and a half north of San Francisco. And one of the things with this piece is that to begin, I chose a landscape that had a curve to it. And it makes a huge difference in terms of the activity of what you put on in order to incorporate that curve. I did a drawing on it with these little knobbies. They're wooden knobbies. And I drew a pathway that made a curve that you can see here. On top of these knobbies, then, I would place a piece of plywood so that I would know where to start building these elements. You see I'm building, and I draw lines that you're seeing here that the cutters cut for me, gingerly trying to avoid these knots, which are always the obnoxious thing. And I keep saying that when my ship comes home, I'm not going to have any more knots in my cedar. But this is what the cutters cut for me, is they follow the lines that I draw for them. This is the piece from the front. It's in an apple orchard. This is the piece from the side view so that you can see a kind of procession. One can actually walk through this. It's about chest high, and it, in a sense, is very dense in the front, and then it unravels as it goes toward the back, so that there's a different kind of activity that occurs there. That's the unraveling. This is a bowl with folds, and my titles are often just titles that feel very documentary, but you get the sense of the scale, and it's actually a bowl. It's hollow on the inside, though some of the walls with the thickness of the structures are about 9 feet thick. This is a piece that is right here in New York City in the Bloomberg building. It's in the lobby of the Bloomberg building. And there's something to me about the sort of insistent corporate minimal look of that architecture that made me want to do something that was almost opposite to it, that was more sensual, that was more inviting. So this is actually copying the floor of that lobby. That is, it has a lean, and often you can't tell that with New York City sidewalks when you're walking them, but there are curves in Manhattan with the landscape. This is the process of gluing the 2x4 pieces of cedar together. It's a very labor-intensive, almost medieval-like, game that I invented for myself that has a lot of sort of sweat bullets in it. But I need that, I wanted that. So this is reverse stacking the pieces that I first screw all of the wood together, then I reverse stack it, and then layer by layer, we put it back together again exactly as it was screwed after having marked it very carefully. So you see this huge spread for something that'll condense itself. I'm putting the graphite on here, and that is comes with very particular visuals. Meaning that only I can do the graphiting, because they're really consequential in making the paste look like what I think I need it to look like. So this is a kind of procession as well, with all of these edges that are the lips of these vessels that are lining up in a way that's kind of erratic, but not. They're obviously of the same family, and usually this hallway is completely crowded. So that this is the other side of that. It's 88 feet, 6 inches long, and it comes out about 5 feet, 4.5 feet, like so. And one of the consequential things for me was that it had a back on this vessel that attached itself very firmly to the wall, that was flat, and from that flatness came this more sort of organic, running, almost a kind of lip. This is a full-scale model of a piece that was in the, it was about 2006 at Madison Square Park Conservancy. It's a park on between 23rd Street and 27th Street. So, but the important thing here is that I submit, this ended up being in resin, but I submit the full-scale model in cedar. I make the entire thing in cedar first, before then it gets cast. Therefore, I feel like I'm in control of the visuals. It gets smothered with, this is a plaster cast on it that's reinforced with the metal. This is at the foundry, it's Walla Walla in the state of Washington. And this pink stuff is rubber that can get very sensitively into very small areas that are very important to me. So it's extremely, it takes great care in getting the surfaces that I gave them, because that's a huge part of how I communicate through these through this artwork. So this is the piece as it is in resin. Same place, Madison Square Park. So you get the idea that it does sort of glorious things with light and the sun. And this is the result of using that pink rubber stuff to cast with. You can actually pull that rubber out from underneath this thing that almost feels like a scale, so that it's quite sensitive. This is in Sweden, an island named Djurn. Sweden. That's a piece that's at the Walton Museum now, and it's a piece that can be actually squeezed onto a ceiling, but it can also be squeezed into a corner or neither one. But you get that sense that it has that possibility, and that's how I built it. I actually built it on the floor against a wall, deliberately making these things, you know, that kind of mobile, you know, that elastic's not the right word, but having that kind of energy or having that kind of grouping. Another piece, horizontal. Now we're at about 3 years. This is about, I made this about 2.5, 3 years ago, heavily horizontal. And I started with this section, which actually I carved out of the inside of a failed bowl, but it had hundreds of shims stuck into it that made it feel as though it had history. I ground it with a grinder you're only supposed to use for metal, giving it a burnt look to increase the fact that it's been kind of used and abused. And then I started by putting 2x4 cedar beams, but it's all 1 by 1 again. I draw on each one of those 2x4s, and I line them up, but I straighten them up as I go back. That's a part of the movement. And a part of the movement is that there's a belly-like structure that tucks itself under as it hits the ground, so you get a sense of a large thing, you know, that goes at a particular pace that's not exactly fast. That's the other side. It tears itself open and it rips out with all of the innards. Close up, so you can see even with the 2x4s that I glued together that there are edges that I leave raw to give the sense of a shift or of a movement, much like what the fish does when it turns its body and the scales shift onto one another. And this is just leaving the cedar, and that's one of the first times that I've done that, completely without any graphite and flat. and giving this creature a kind of pattern. I don't even want to call it a creature because I don't want to identify it in any way. And I prefer that the people that look at it make their own metaphors. This is a piece now that's at Storm King. I'm building it here. That's the piece. And I had this fantasy, not that it really counts to anybody else, but pod pachom in Polish is underneath your armpit, and that's where the bread is carried, that's where the babies are carried, that there's an intimate place. And I thought I would frame this beautiful colonnade of trees with this kind of frame. And you have to line yourself up in order to see it. And it really doesn't matter to anybody else, but it was a motivating force. But even a greater one, I think, was a kind of vulnerability that I was after. It's the first time I made one of my works touch the ground, and it would touch the ground with a darker, harder, thinner structure appended to something that was much more, tumorish and much thicker, that appended itself to the main body. And that you get a sense of scale here. All cedar with a little bit of graphite. And at this stage now, and it's a little, this is not long ago, I made this about 2 years ago, I want the cedar, I constantly have this desire to make it look lighter, to make it feel like a fabric, to make it feel like a fabric that's even being unfurled, or sort of titillated with the wind, but not that this is it, but that I have that desire. Another piece that I made fairly recently, and these two are kind of in my head, marks of distinction, and she has on her back a very powerfully strong back, and that it's very, very flat. That is that it has the 2x4s that are lined up on top of one another. But in my head, it gives her then the power to be able to hold on to and sort of go as kind of wayward as she does on both of her sides. And I have a close-up of this. You see, this is the bare, raw wood that these marks are covering on either side. This is my studio. Or it's part of my studio, it changes constantly. A small plate. This is called oddychanka, and in Polish it's it makes reference to a female breathing. It's about 10 feet wide. It's just essentially a drawing that I cut out, a line drawing. This is at the Decortiva Museum. It's made out of resin as well. Shovels. I come from a long line of Polish and Ukrainian peasant farmers. And there's a thing that I have for shovels. I've made many, many. I don't even want to have a thing for shovels, but it seems to be there and it seems to want to assert itself. So this is a flat back that I give, and I haven't done this very often. This is the only time I've done it, in fact. But what's important here is that you see it sucking itself out of its ground. There's another self that comes out first because there's almost like a cavern in back of it. And then there's something else that appears at the top, it's whatever. It's another kind of look or another. But they're obviously both related. It's just a transition. And these are marks from the sneakers that we stepped on top in order to make this piece. So I chose some of those patterns and cut them out. Again, in an effort to just make it lighter. And this is another shovel that again has a one kind of self coming out and then another that it ends up being. But look at how voluptuous this is. All done with a circular saw, which is only meant to cut, straight, that's it. And we do this by nibbling. You know, hold on to the 4x4 and we nibble with that circular saw, almost like an animal nibbles. And if you have enough straight nibbles with that saw, because that's all it can do is cut straight surfaces, you get a curve. And that's what I'm after. And to this day, I'm very grateful that I have never taken a woodworking course. A wall. But the wall begins with something that's more minced, and then it goes to things that are sort of more swollen as it goes down, and that there's a kind of interaction between the grid of the wood. You see the grid, but then interaction with all of these organic structures that get superimposed. You know, so they're kind of, you know, have to live with one another, but they're quite different. That's my drawing on a piece that's going to be added onto here. But you see these cuts, these are very different from any other cuts that I've made. They're corrugated. Again, this is nibbling to make these things. And I think I could get into the World Book of Records for the kind of things that we do with a circular saw. And I think the cedar, too, never imagined it would be put through such grief. So here's the drawing that I make for the cutters to cut. See, and I'm actually mimicking at the beginning here, and then I make it move by making something else on the other side of the 4x4. So this is all about movement. And I found this in my studio, and it's what it actually inspired. It was sort of begging me to, you know, make it do something, you know, keep it going. This is the beginning of a structure. This is all going to be process again. It's a bowl. It's an empty bowl. And I look at it, and I deliberately made one fold that was kind of, you know, dramatic in it. And then I carve it up. I carve it, you know, I never ever use a chainsaw, but I've carved this with a Sawzall. And then I put shims into it. You see, I open it up. Because the process of destruction is often as marvelous as that which gets constructed. So you see, that's my shims. It's lying down now. I get rid of some of these shims because they're too aggressive. That's the piece now. I added an end to it and a beginning to it. I covered the blue so that wouldn't get any graphite, but I'm gently graphiting the rest of it. That's the piece. And that's kind of its beginning, and that's kind of its end. And there's a struggle in that it lifts its body up and then down again. The only places it touches the floor is here and here. And it's my dream one, you know, to get rid of this. But I haven't been able to do it yet, but I'm determined that I will. That's its front. That's kind of its head. This is a piece that's a flat piece, it's a relief piece. Ignore the black, because I put this on after, but I just want you to see what happened with this. 33 sections that I cut out, 33 sections. They're rectangles out of this relief. I put abaca paper on top. I coaxed it in with a brush and a little bit of archival glue. And we're taking the paper off now. This is after a few days of drying. Abaca paper is made from a stem of a very rare banana from the Philippines that gets whipped into a pulp. You see, then we put this together, all of the separate parts together, and reinforce the frames. So you get the sense of the scale. And this beautiful color that happened was what the cedar released. That's the color, the juices that came from the cedar itself. And I was so wanting of this color again, or of this whole pattern, because I hate color. I mean, I wear nothing but black, but this color is so doable in my head. It's so sort of psychologically easy for me to take in. That I made another, I took those same forms and again put the abaca paper over it. It's wet when you do that. And it turned all white. It would only give this color once. It only gave me that one opportunity to do it. So this is a piece that'll end up being glass. I made this in cedar first to see if I can have some sort of pattern to go by. This is in Murano. I'm setting this up on the floor, and I take each one of those bars of glass and I ram it into a wheel that's diamond studded that carves into each of these bars. And my favorite part, is what happens in the matte areas. I hate shine too, but I let it shine just a little bit because that's all glass is about, actually, is the kind of sheen that you get from its surfaces. And this is in Venice at Palazzo Franchetti. My drawing again, the piece growing. This is my cutter. They're the princes of my studio. It grows. This is Ona. The one that's at the Barclay Center. I'm making the full-scale model out of cedar. Look at how convoluted this profile is of the bottom portion of Ona. And it needed to be that way because around here is where her belly is. And I made the belly for, nobody should even knows this or realize, because it's not literally a belly, but it's a place that feels awfully good to the touch. So this is what it looked like, and I had to have it in two sections because this is the bottom of it and this is the top of it, because my studio can't fit that height. This is what it is in the Foundry, Polish Artworks, upstate New York. This is the bronze. This is the cedar. Look how abused this poor thing looks. So I'm going to see if I can excavate it somehow. But this is only half of it, not even half of it. That's the bronze. The other side of that same piece. This is this is sand casting. You see, so they cast it around a point like this. This is my piece on the inside, so they can then take it out and cast it. It's cheaper and it has a slightly more grainy surface, which I like. This is one of the donuts of that bowl. They're actually stacked, hollow shapes like this. This is the bronze, obviously. Bronze, look at the welding on the inside. Very organic, really difficult to fit. That's the cedar piece. This is the top of the bronze. You can see the top section. This is my patina. I felt like an impressionist painter. I wanted to liven it up. That's the patina. That's the bowl. We're doing the patina on it. And you see here a stainless steel flat structure so that birds don't get caught in there or anything. Well, nothing will get caught there. So this is how she came. This is Ona. This is at the Barclay Center. How she came, and her folds, I was so grateful, actually were in keeping with how gravity might pull those folds. That's the piece installed. This is the cutting of the side grain of the cedar that looks very different from the cutting that happens when you use the front grains. This is still the side grain, but you can get a sense of the patina and that it dies down after it gets waxed, so it's not quite so flighty and crazy. This is the finished piece again. So you can see how inviting it is for the hand, for the touch. It's actually very smooth and almost silky in the way it feels. And I just wanted to say something that I never read, but you know, my talk, but this is like a 3-minute, 2-minute thing. So you can still hear me okay? Okay, it was 4:30 in the morning of October 9th in the year 2000. When the phone in our home rang, my husband, Paul Greengard, dazed from being woken up from a deep sleep, mumbled something akin to, "What idiot could be calling me at this hour in the morning?" Our daughter, who was sleeping in the adjacent bedroom, answered the phone. Paul, with his driving curiosity, picked up the phone and listened to our daughter say, "Do you really want me to wake him up?" Our daughter said. "It's 4:30 in the morning." While Paul was still listening to the phone, a man's voice answered, "This is Hans Jorenvall of the Karolinska Institute in Stockholm. Please, I would like to speak to Paul Greengard." Which Paul immediately replied, "I'm awake, I'm awake." It was during our short walk that morning, between being accosted by the photographing press from Paul's laboratory to this auditorium, that we decided we would leave the Nobel Prize money to women scientists doing the best biomedical research in the world. We would establish this prize in honor of Paul's mother, who died giving him birth. From the time Paul Greengard was born, his mother's existence was kept from him until he was 21 years old. We know little about her. A photo which we thought was of Pearl Meister we placed in a position so that she could witness our marriage 35 years ago. The photograph turned out to be another woman. There were no photographs, in other words, of his mother. We were told Pearl Meister was beautiful, had thick dark hair, took many baths, and read a lot. She was a secretary. In the days of the 1920s, a woman could basically be a teacher, a nurse, or a secretary. This, plus Paul's long history as a scientist, witnessing discrimination against women, enabled us to make the decision about the prize with such ease. And, of course, I need to make clear that I'm but a small part of this, as it is Paul's scientific brilliance that enabled us to establish this award, along with many others connected to Rockefeller University who contributed to it. Pearl Meister was in some way concretized, in a way living another life, floating in the highest and most pure of atmospheres at Rockefeller University. This was tremendously healing to Paul. In addition to singing the praises to this university, I want to thank Marnie Amhoff and her team of smart, so beautiful women who have shepherded this prize so well for the past 10 years. Shortly after having received the Pearl Meister Greengard Award, Elizabeth Blackburn and Carol Greider received the Nobel Prize. I have had the privilege of sitting through these awards for the last 10 years and have been deeply moved by the astonishing creative accomplishments of these women scientists. Thank you.
Thank you, Ursula, for those beautiful words, and thank you for that inspiring introduction to your remarkable work. I think we'd like to give you a round of applause for that as well. I also want to say how deeply grateful we are to you for your role in creating this award, as well as your contribution to the Rockefeller community at large. And now it's my pleasure to introduce Dr. Huda Zoghbi, our honoree this evening. Huda began her medical education at the American University in Beirut, but her studies were interrupted in 1975 by the Lebanese Civil War. She came to the United States for what she believed would be a temporary visit with relatives, but as the conflict escalated, it became clear that she would need to complete her studies abroad. She was admitted to Meharry Medical College in Nashville. Following Meharry, she accepted a residency at Baylor College of Medicine in Houston. Huda found herself drawn to neurology and the opportunities it provided to combine patient care with analytical problem solving. Her interest in rare inherited neurological diseases led her to undertake training in molecular genetics, and thus began a remarkable career in research neurogenetics, a field that she helped to shape as we know it today. Huda's investigations of rare diseases have turned out to be have had far-reaching implications for medicine, and in her own words, quote, "By researching diseases that affect only a few people, we might solve others that affect many." Huda's enthusiasm for her work has made her a cherished presence at Baylor, where she is now the Ralph D. Feigen Professor in the Department of Pediatrics, Molecular and Human Genetics, and Neuroscience. She's also the director of the Jan and Dan Duncan Neurological Research Institute at Texas Children's Hospital and an investigator of the Howard Hughes Medical Institute. A member of the National Academy of Sciences and its Institute of Medicine, Huda has received numerous other awards, including the Gruber Prize in Neuroscience, the Vilcek Prize for Biomedical Research, and the Bristol-Myers Squibb Neuroscience Distinguished Achievement Award. We're also delighted that Huda's husband, Dr. William Zoghbi, and her son and daughter-in-law, Anthony and Zena, are able to be with us this evening. Now, in a few minutes, I will talk with Huda about her career in science, but first, please join me in welcoming her to the stage. Ursula, will you also please join us? So Huda and Ursula. So it's my pleasure to read excerpts from the citation that's being presented to Huda Zoghbi this evening. And I quote, "You are being honored for your groundbreaking discoveries in neurogenetics, which have broadened our understanding of inborn neurological disorders and led to key insights into how the nervous system develops and functions. Through your compassion for your patients and their families, coupled with a determined pursuit of knowledge, you have provided new hope for the treatment of a range of intractable diseases. Today, in presenting you with the 2013 Meister Greengard Prize, we recognize and thank you for your pioneering achievements, your dedication to the progress of biomedical science, and your unwavering devotion to the betterment of humanity." And now, Ursula will read a short statement as well.
It is my honor this evening to present to Dr. Huda Zoghbi the Meister Greengard Award, an international award recognizing outstanding women in biomedical science. On behalf of the distinguished jury of the Meister Greengard Prize and all of us here today, we congratulate you on this award.
Thank you so much.
I knew I will be speechless after this, so I wrote a few comments ahead of time. I just want to say how deeply honored and humbled I am to get this award and to have it at Rockefeller University, no less, one of the institutions I truly admire, and half of the scientists are my closest friends, so it's truly a great honor. Ursula, what an honor to be introduced by you, a true treasure, whose art stands tall, beautiful, and so unique. It talks to me. It's really beautiful. It was amazing. I'm grateful to your generosity and for what you and Paul have done to increase the recognition of women scientists. Paul has been one of my scientific heroes, and he has been an inspiration. He doesn't know it, but for many years, starting from that time, he took me to that small Italian restaurant. I will never forget that dinner when we talked all night long about science and problems we want to solve. I've always wished for more time, and I was always sad when our meeting ended because I felt there's so much more we could talk about. To be selected out of all the great and accomplished female scientists is far beyond anything I've ever imagined. I consider myself extremely lucky for being rewarded for doing what I love to do, research, where every day brings its own rewards. It goes without saying that I'm indebted to my mentors, collaborators, and trainees. I'm grateful to 3 particular mentors: Ralph Feigen, who recruited me to pediatrics at Baylor, and he taught me clinical scholarship and became my second father. I'm also grateful to Marv Fishman, a neurologist, who convinced me I should not do cardiology and switch to neurology. When I decided to do training and research, Art Baudet, one of the finest geneticists in the country, took me into his lab and taught me how to be a scientist. I owe him more than words can express. I've been fortunate to have many collaborators, but I'd like to mention 3, because I've worked with them for decades and they're really part of my family: Harry Orr, Hugo Bellin, and Juan Botas. They've been great friends and comrades in our shared quest to understand neurobiology. And of course, my wonderful trainees, the students and the fellows who took many risks, who followed some crazy ideas and put them to action, and worked tirelessly and with passion. Most importantly, I want to thank my husband, William. Since our first medical meeting in medical school, from meeting the demands of 2 busy careers to raising 2 wonderful children, he has always been my one stress-free zone. I tell him when I'm with him, that's the stress-free zone I feel. He has supported me with unconditional love and unfailing good humor. Our life together has made my career and this award possible. Thank you.
Thank you, Huda. And now we will have a conversation and also invite the audience to join us a little later. And I wonder if we could perhaps start by having you tell us first how you decided to go into medicine and then how you decided to switch over to research.
So I actually did not want to go into medicine. The truth is I wanted to do English literature. But my mother insisted I should do medicine. And she said writing could be my hobby. And when I said no, she cried and finally said, okay, I'll do medicine. And that was really how it happened. And I think I got into medical school at the American University of Beirut because of my high grades in literature. So it all paid off. It all paid off.
That's right.
Yes.
But you didn't look back.
I did not look back, and I enjoyed every minute for it. I have to say, my mother knew I was good at biology, and she thought, you like it, and you will see you will enjoy it. And that was her insistence.
But then you moved, well, you staying grounded in medicine, you then moved towards research also. How did that happen?
Right. So my move to research was really totally driven by seeing patients with devastating neurological problems. And we have to go back to now the early 1980s when we didn't have any way to diagnose many of these patients, but we knew that these are genetic problems because we would see 2 children in a family or a parent-child affected. And it felt miserable to tell them this will happen again, maybe 1 in 2 or maybe 1 in 4 pregnancies will be affected, but we could do nothing for them. And that's when I decided I really need to go do research and figure at least some of these genetic disorders, maybe we can do something about them. And I talked to Art Baudet, who I think was very brave to take in a fellow who hardly has done any research, and he took me in.
I understand that you entered his lab on a handshake.
Absolutely.
Yes. Could that happen again today?
No, a few things happened. He did do one thing. He sat me down, he gave me an exam. I think he wanted to establish what's, the ground rules. So he sat me down, he gave me a big genetics exam. And then he said, great, you can start. So that was, you're right, that doesn't happen again. And the other thing that happened that doesn't happen again, and I keep telling the NIH this, once I decided to join his lab, he said, go write a physician scientist award, the KO8. I said, great. He gave me the instructions and I decided on the project and I took Maniatis home. And every time I have an idea, I will look up in Maniatis, how do they do it?
This is the big textbook of molecular biology.
Right, this is a textbook, just a protocol, how you do experiments. And I would think, I would read how they do them and I would write it and then I'll show it to Art the next day. Does that sound right? So I had no experience, no data whatsoever. And I started in the lab with the first day of funding of the KO8. And I think this is for people who really are serious. I wish they would do that again, but nowadays, unfortunately.
Now you have to have years and years of data before.
Years. Yeah. Right.
So, well, we'll come back to the research later. I know that people want to hear more about that, but I do have to ask you, Meharry Medical College, how did you decide to do that? And also tell us about going there and also your impressions moving from Beirut to Nashville. It's a legendary city in the US. What were your impressions when you first got here?
So my first entry to the States was to Austin, Texas, because that's where my sister was. And it was July 4th, 1976, correct. So it was the bicentennial. And my first impression was there was war in Texas because I'd left bombs in Lebanon and I did not recognize that fireworks were celebratory. So the first thing that happened, these sounds, and I really cried. It was truly PTSD. Now we know it's post-traumatic stress disorder. I started crying when I heard that. Anyway, that was in July, and we were hoping to go back to Lebanon in October when medical school started. October came and we could not return. And that's when I realized I have to find a medical school. So I started reaching out to medical schools, and really nobody will take a transfer student in October, 2 months late, and even medical school transfer is not so easy. So I was pretty upset about it, and a friend of the family said, come to Nashville. So I went to Nashville, and he said, I'm going to take you to Vanderbilt. So I went to Vanderbilt, and they said, no, school started 2 months ago, we will not take you, but try Meharry. So I went to Meharry, and they looked at my grades and said, we'll take you. We don't care that school started 2 months ago, we'll take you. So that's how I ended up there.
So another example of flexibility that is pretty rare these days. It would be harder to achieve that now.
Absolutely. So I have to say, Nashville was a true culture shock for me, because the first thing that happened, nobody understood me, and I didn't understand anyone. I didn't realize words could have so many syllables coming from Beirut, and they could not understand my accent. So it was a little bit of a challenge. The food, there was no yogurt in Nashville, and in Lebanon you eat yogurt every day. So there were little things like that that were difficult. And then in the school, I also felt very different. For one thing, everybody talked about sports and I couldn't relate, and so on and so forth. So it was really a big adaptation, starting 2 months late, family away, bombs falling in Lebanon, you can't talk often. So I was really worried about them. Had just met William, heartbroken that I left the nicest person I met back in Lebanon, and I'm here. So there were so many, it was a very, very rough year. Having to catch up the 2 months of medical school and having to really adapt to a culture very foreign. I've never lived outside home. At the university, we lived in bunkers basically because of the war, but up till then I was still at home. So it was a really big
But you adapted.
I adapted. It was a milestone year. Every time I faced a challenge, I always thought, when experiments didn't work, I always thought, it's not as bad as my first year in the United States, right?
And so then you went back to Texas, where they have fireworks.
That's right.
And tell us a little bit about you mentioned the patient, seeing patients and being inspired. I understand there's one patient in particular who really captured your heart and your imagination that drove you into a line of research. Maybe you can tell us about that patient and how that got you into research, and tell us about your research program.
Sure. So really the defining moment that convinced me I should go into research was seeing a 5-year-old brought in by her father, who's a professor of economics, and her mother, because over 2 years they saw her change completely. She was a healthy infant born doing everything a little girl would do, including using her hands, feeding herself, singing a few, to some rhymes, and saying a few words. And then gradually she fell silent. She stopped greeting her dad at the door and became autistic, lost the ability to use her hands, and instead will constantly wring her hands. And over 2 to 3 years, they watched her lose all the skills she learned and become stiff and unbalanced and having seizures. And it was quite a devastating situation. So they brought her in, and I was just struck by her picture, because I felt that to be so normal and to deteriorate, but yet her scans and everything did not show evidence of degeneration. So I couldn't quite understand. Her brain development obviously must have been normal, and then something happened. And in neurology, we don't see that. We see either somebody born with parts of the brains missing or cell types or so on lacking, or degenerative. So that's what intrigued me. But it was one case, and I had read about the disorder that happened to read about.
Rett syndrome, is it?
Read about the disorder. It was the first time described in the American literature, in the English literature that same week. And it's called Rett syndrome, described by an Austrian pediatrician, but reported by Swedish pediatric neurologists who recognized the syndrome and reported the first 35 cases. So I was really struck, read the 35 cases and her case, and it was quite all girls, and I thought this is really intriguing, but it stayed there. A week later, I'm in the clinic, and I choose to see a patient with cerebral palsy, and I thought those are always an unknown, and I can solve this puzzle. And a 12-year-old walks into the office wringing her hand. At that point, nobody in the US has seen a patient with Rett syndrome. So I see my first patient 1 week, and I see a second one a week later, and I immediately recognized she had Rett. And immediately I ran to the clinic volunteers and asked them to find me every chart of a female with, I listed the features, and they came back with 35 charts, and I found 7 more. So within about a span of a few weeks, I had a handful of Rett syndrome patients, and I saw them all, and I was really intrigued and decided to do some clinical studies on them, published the first paper on them in the New England Journal of Medicine, and then all of them descended. Within a short time, we had 200 patients in Houston. And you see them, girl after girl, the same story. First normal, and then losing milestones, and my oldest at the time was 35 years old, and they were all girls. So I decided I'm going to clone the gene for this disease, and I went to Art and I told him I want to clone this gene, and he said, great, show me I said, I have 200 patients, show me the families, and they're all sporadic cases. And he said, no way, pick another problem. And that's how
it would be too difficult,
in
those days.
In those days, you couldn't map it, so you couldn't really, the genome wasn't sequenced, there were no maps of chromosomes, so he said, pick another problem, and that's how I picked the neurodegenerative disease that I study, which was really clearly inherited, a dominantly inherited disease. We had a big family in Montgomery, Texas, and that disease affects balance and coordination. Adults, typically in their 30s or 40s, will lose their balance and coordination, and gradually they'll lose all motor activity to the point they can't even swallow and breathe in a coordinated way, and within 10 to 15 years will die. And I thought that's really very interesting, because why does it take 35 years to show degeneration? Why is it that only one mutant gene is enough to cause the disease? And I thought this is very similar to another disease called Huntington disease, but being a new person who doesn't know how to do any experiment yet, I didn't want to work on Huntington, where there are many big labs all over the country working on it. I thought this would be very similar, mechanistically maybe, but at least it's a unique disease, and that's how I studied the ataxia.
You found a niche where you could actually make progress without worrying about the competition.
Correct.
But then, and you ultimately went ahead and cloned that gene, discovered it, and in fact tomorrow you're going to be telling us about some of your advances in understanding how it causes the degeneration.
Correct.
Is that right? So, but then you decided to go back to Rett syndrome.
So while I'm working on the ataxia, I'm still collecting patients, and I'm still pursuing Rett syndrome, doing different experiments to really try to wrap my head around this disease, all females. I figured it must be on the X chromosome, because I couldn't really imagine a way to explain it without it being on the X. And the reason I thought it's on the X, because females have 2 X chromosomes, whereas males have only 1. And I figured if these mutations happen on the X, the female has another X in 50% of her cells that are healthy, and the 50%, because females are mosaic. So every female in this room, although she has 2 X chromosomes, but in every cell, only one of the X's is active. And this is because nature wants males and females to be equal, and just like Paul and Ursula in science. And in males, you have 1 X, so in every cell, you have that 1 X active. In the females, you have 2 X's, and to have both of them active, then the females will have a lot more genes, so you always inactivate one of the X's. So I thought in the female, 50% of the cell with a normal X, 50 with a mutant, will explain why a female will make it, whereas a male who only has that 1 X, if it's mutated, he'll be dead probably. So that was the model I built.
Interspersion of of
Right.
healthy and unhealthy cells.
Right. Interspersion of healthy and healthy cells will make them live, but if you only have unhealthy cells, maybe they will not live. So with that model, I started looking for the gene on the X chromosome. And it was really marching down the X until I got lucky and I found two sisters who shared a mother but had different fathers. And then I used them to see which portions of the X they actually share. And 10 years later, or 7 years later, another pairs arrived. So I was able to narrow it down from the whole X chromosome, which is almost like looking at the whole East Coast in the United States, to New York. Now I'm searching for this one mutation in New York. So we're marching down the X gene by gene sequencing until we finally found it. So it took 16 years.
16 years. So that persistence that you learned in Mahari served you well later in your career. But in pursuing those two projects side by side, does that help? Well, when you're out in the desert, walking through the desert on Rett syndrome?
Right. It helped tremendously because at least with the ataxia project, where I had a large family where I can use the tools of genetics to slowly map the gene, I was getting some small step rewards, whereas with Rett syndrome, imagine marching down the X chromosome and sequencing gene by gene. It's normal, it's normal. It was 15 and a half years of negative results, so it was pretty depressing.
Yeah.
So you need that other project that sort of kept me going.
But at the same time, that persistence is essential to get to the root of the problem.
Right.
So then you have the genes in hand. Now, I know it's complicated and quite technical, but could you explain maybe just a few words what you discovered by discovering the Rett syndrome gene? Mm-hmm. What goes wrong in the brains of these girls? And maybe a few words also about the degeneration.
Sure. So to tell you a little bit what we learned after discovering the gene, it turns out the gene makes a protein that binds the DNA. And in our DNA, everybody knows the 4 bases of DNA: adenine, thymine, guanine, cytosine. But the cytosine sometimes can be modified, and this protein can bind it when the cytosine is modified. And that modification allows the protein, when it binds, to change sometimes how genes are expressed in our cells, whether they're up or they're down. So I'm talking to you today. A part of your brain, the hippocampus, is trying to capture this conversation. You're trying to remember some things from this conversation. So your cells are going to be very busy making new proteins and expressing new genes so that you can consolidate some of this information and help the synapses form this information. That's when this protein comes into action. You need this protein to adapt the cells to a new change so that it can acquire information, or when you sit down today to eat dinner, you need to know when to stop dinner. The cells in the hypothalamus that will respond to how much food you've taken, they respond because this protein is active in them. So slowly, by studying this protein in different cells in the brain, using mouse model that we engineer to take it away and see what happens, we learn about its function in many brain cells. So it turns out to be important for practically all brain cells. It's sort of the master conductor in all brain cells, and it tells genes when to express themselves and when to be quiet. And that's important because that's the only way neurons learn how to respond during challenges and different activities. And that's why the poor Rett syndrome girls have all the symptoms of every neurologic disease. I didn't tell you all the other symptoms. When they get older, they have Parkinson, they have dystonia. So they have pretty much every symptom of disease. Now, this is when you inactivate the protein completely. What we also learned is that if you just slightly inactivate it, which means you cause a mutation that doesn't totally kill the protein, but make it milder, then you can start having symptoms like bipolar disorder or juvenile schizophrenia or autism. So it's really a very important protein. Every way you change it, too little, too much, you make it overactive, it's also bad for the brain. So that's what we've learned, and we really only learned that from going into the animal models.
So does this knowledge point to potential therapeutic avenues for these girls or potentially other individuals who carry mutations?
So what we've learned since the gene discovery is all the changes in the brain and all the chemicals in the brain that are really essential for normal brain function that are decreased when this protein is gone. And some of those are so important, and there are medications that can potentiate them. So what we're doing now, we're testing some of these medications in the animal models to see if they would improve some of the symptoms, and then one can move to human patients. So it's opening up really potential pathway to investigate therapies.
So it provides the tools to try to get at them, although you aren't there yet.
No, not yet.
And on the degeneration side, any brief insights you can provide us with?
So on the degeneration side, we started with a very, very rare disorder. I told you it's an inherited balance disorder. Its name is spinocerebellar ataxia. We call it SCA1 for short. And it happens in 1 in 100,000 people. So it's really, really rare. But what we've learned from finding the gene is we learned that the mutation caused the protein to accumulate in the brain cells. And having a protein, normally our brain cells are used to having just the right amount of proteins, and we're learning this now more and more. So it doesn't tolerate having even 50% more or 50% less for many proteins. It doesn't tolerate that. So in this case, this mutation makes the protein accumulate a little bit more, and when it accumulates a little bit more with time, these brain cells are experiencing a lot of changes in the chemicals they make and the proteins they make to where it becomes toxic and they eventually die. So our studies led us to really discover that the level of the protein matters, and if we can find ways to decrease the level of that protein, we discover we can rescue the disease. A few things we learned that were very, very exciting. First thing we learned, and I should mention my work on SCA1 is in collaboration with Harry Orr. What we learned is when you have this mutant protein in a mouse and the mouse gets sick and has all the features of the human disease, if somehow you can reduce the protein or decrease it or eliminate the mutant protein, the brain cells recover. What was really a surprise, we had no idea that very sick brain cells where the animals is losing its balance and falling all over the place, you now turn the mutant protein away, they make new branches and they're healthy. Of course, if they died, it's too late. But I think this gives us hope, because I think when we think about diseases like Parkinson and Alzheimer, there are many, many symptoms, very subtle symptoms that happen very early on where the brain cells are still there. And I think if we can intervene at that point, we can reverse even some of those early symptoms. So that's one thing that to me was startling when
the reversibility.
The reversibility when we learned that. Now, having discovered that, we've identified ways to find, we're trying to find drugs, chemicals, pharmaceuticals, that will lower the level of this mutant protein. And we established a strategy that I'll be talking about tomorrow that genetically will lead us to things that you can actually inhibit using pharmaceutical drugs. And we recently discovered one such inhibitor, and we're now trying to really make it turn into a medicine. As we talked during the reception, first you use an inhibitor that's maybe a chemical, not safe to be putting in human, but you can study it and learn the mechanism with that inhibitor and eventually work with somebody who knows how to do medicinal chemistry and move it into a medicine, we hope. And that's one important thing we learned from the study of this disease. But the other thing is we learned that many of the things we discover for this disease really apply to Alzheimer, Parkinson's disease, Huntington disease, and other degenerative diseases. So these diseases inform each other, and that's really what's also valuable. We learn from each other and we inform each other.
That's great. Well, I want to open it up to questions, but maybe I can ask you one more questions and get back to move away from the hardcore science. You have just received a prize for women scientists. Are women scientists different?
I think all my male colleagues told me I should not work on Rett syndrome. They all thought I should quit, it's going nowhere, and there were not many female scientists for me to discuss the project with them. So I would only talk to my male colleagues, and they all said, don't do it, it's worthless. So I stopped telling them I work on it. There were 7 years of silence, 6 or 7 years of silence. I didn't tell anybody anymore I work on Rett except the one fellow working on it. Because I was really sort of intimidated and feel everybody thought I was crazy to work on it.
So they thought that it was just too risky to...
They thought there's no way this is a genetic disorder. This is a sporadic disorder. It's one in a family. How do you know it's genetic? And you're wasting your time and you're not going to get there. So they felt it's a project that's not going to go anywhere. So that gets me to the point, the slight difference in women's health. You have a gut feeling, and if we have that gut feeling, at least for me, if I have a gut feeling about something, I stick with it. No matter how challenging, I will not quit till I see it through. And I think my colleagues were much more, where's the hypothesis here? Where are the data to support the hypothesis this is on the X? So they're very, very strict in that sense. And I think that little gut feeling for me that let me stick with it. I don't know. That's the only thing I could think about, maybe.
Great. Well, I think we have some roving microphones, and do we have questions from the audience?
I congratulate you on the progress you made in this rare disease, and I recognize the importance of the methodology that you've developed in terms of recognizing the effect of a protein over many years. But there, as you well know, there are diseases like Alzheimer's and Parkinson's that affect millions of people, and I'm wondering to what extent you feel you can apply the lessons you've learned in these more widespread diseases.
Right. I think Bob's question is very important. What we've learned from the ataxia study, how can we apply it to Parkinson and Alzheimer's disease, which affect millions and millions of people? And that's exactly where we are today, Bob. It's a right-on question. So in Alzheimer's disease, there are certain proteins that we know accumulate. We also know that the dosage of a protein, sometimes when it's increased, it can cause Alzheimer's disease. I would give the example of the amyloid precursor protein. It's a protein that if there's a duplication of it in the genome, so if a person has one extra copy, they will have dementia. In Down syndrome, this protein, there's an extra copy of it because Down syndrome patients have 3 copies of chromosome 21, and this protein is on chromosome 21. And Down syndrome patients get this disease, Alzheimer's, in their 30s and 40s. In Parkinson, there's a protein called synuclein, which we find at autopsies that it's in the tissue of many patients who die from Parkinson's disease. We see accumulations of synuclein. And it turns out there's some human patients who have an extra copy of this gene, so they make 50%, sometimes 100% more of that protein. So that principle that having some proteins, having too much of them in the cells, is truly driving many other neurodegenerative diseases. And the same strategy we're using to discover this molecule that I was mentioning to Mark for SCA1, we can now, and we are actually, thanks to the Belfer Consortium in Houston, Texas, applying it to APP protein and tau protein and other neurodegenerative diseases.
I'm curious if your love of, how your love of English literature might affect how you do science.
So I think it does affect it because if you happen to pick one of my papers, and I had an argument with a Rockefeller colleague, I'll tell you about it in a second, they read like a story. And Nat Hines, who's a very dear colleague, and he's here at Rockefeller University, he told me, you write every paper of your students. I said, no, I don't write them. I teach them how to write them. We go back and forth many. He couldn't believe me. I said, I promise. He said, I don't believe it. I said, why not? He said, because every one of them has a beginning, a middle, and an end. It tells a story. And how do you do that? I said, well, that's what I try to teach them, because that's the only way I know how to tell something, is to tell a story. And I think that's part of it that satisfies my desire for writing, is telling stories of science.
How do you relate mathematics to your scientific inquiries? As you know, many scientists now they think that mathematics is the basis of all being. Mm-hmm. And even with philosophical thinking and cognitive functions, the brain, where they're going into nano levels now, right. It's all based on mathematics. And quantum mechanics, particle physics, it's based on mathematics. So, in other words, what do you think about mathematics as the basis of all being in the universe?
I totally agree with you that mathematics is very important. In fact, if you were to ask me, that you have a break, and for a year you can go back to school. What classes will you pick? It'll be mathematics, maybe class of 2 of physics, but mostly mathematics, because I think it is very important, and I think the lack of integration of much deeper mathematics in biology is probably keeping us from discovering a lot more about our genome at a much larger scale level. We only right now try to understand genes within a certain context with the limitations of what computations we can do with the large sets of data we generate. But there are a lot more data as genomes get sequenced and as we begin to assign phenotypes or features that humans have to what variants they have in the genomes, the variations and the combinatorial the different changes that will happen at the same time that may explain a clinical features or a behavior or a risk factor are really quite complex. And I think the mathematics to solve it all are not there yet, or if they are there, we haven't brought them into the biological realm to solve them with the same extent.
Dr. Zoghbi, can you share with us what are the main obstacles for a cure in Reds?
So the biggest obstacle for a cure in Rett is that this protein is so critical, does so many functions in so many cells that you're going to have to find a chemical that will replace it. And I see a chemical because gene therapy is really not an option for this protein. I didn't talk about the discovery we made that if you have twice as much of this protein, you get another progressive and lethal neurological syndrome. So we call it the Goldilocks protein. You have to have it just right in the brain. So gene therapy is not an option. You have a protein that's essential for every brain cell. How are you going to find who's going to substitute for it? And that's really the biggest obstacle. So what we're trying to do is find the 5 or 6 most major changes in the brain that will explain as many symptoms as possible. I'll give you an example. When the girls get Parkinsonism and you give them L-dopa, they have lower dopamine, they do slightly better. They're very anxious. If you give them some drugs that help anxiety, they do slightly better. And the key is to find all these chemical alterations we're trying to do that through genetics to find another protein that's more druggable that can substitute for this protein.
Well, we'll continue at the reception. People can ask you questions, but before we close, maybe I can ask you one more thing. 16 years to get the Rett protein. With today's technology, whole genome sequencing, how long would it take you if you were starting today and you had 200 to 200 patients?
So with 200 patients today, from the time I submit the blood sample to our genome sequencing center and get the results back, it could be done in 6 to 8 weeks.
So instead of 16 years, 6 to 8 weeks, and that's what the technology has done for us.
Absolutely.
Just in the past 2 decades.
That's right.
So it's really remarkable, and it illustrates why today there's an explosion of discoveries of these rare disorders, of which you were a pioneer. So I'd like to thank Huda first and foremost again. But also a very, very special thanks to Ursula for her very moving speech. And of course to Paul and Ursula for making this possible. Thank you very much.